Can we look at a heavy chain?
Yeah.
Because that's got the D region.
So here's the heavy chain.
How'd you get that so quick?
So I just clicked over here. So when I had searched for the barcode, it provided me the heavy and the kappa.
Oh,
So
I see. So
I'm just clicking between the two.
Oh, okay.
So here's the heavy.
All right, okay.
So
Just want to make sure we
So
can.
again, you've got your start here.
Right.
You have your 5' UTR. Looks like a couple of mismatched bases here. Deletion of 18 bases, some mismatches, and I again don't see any sort of stop introduced at the moment.
Well, that's good. I mean, there shouldn't be a stop.
We hope not.
Is there a way to tell? I know there's. How long is that? How many base pairs?
That's a good question.
Because actually, we got to make this thing. You know, we don't have the amino. We don't have the nucleotide.
Mm-hmm.
We don't have the clone. You know.
I think that would be right there on the front. I don't see it. I mean, I see that there was 24 UMI counts of that particular barcode. There was 13, almost 1,400 reads that aligned across that.
Unique molecular or unique
Molecular
UMI.
index. Yeah.
Unique, so 24. Is that right?
Mm-hmm.
So sequence it 24 times. They found that gene 24 times.
Over 1,400 reads. Yeah, that's what I'm. Isn't that how that's reported? UMI is a unique molecular index that's put on each cell. So yes.
So I found that gene in a cell
Right.
24. Just so 24 times. That's good. I mean, it's
only-
not sky high. But
I don't see a thing inside VLoop that says how long is that sequence, but I guess I could come up here and count the number with inside Excel. You can say count the number of letters, or put it in Word and then say word count.
But you
It'll
got
spit that out.
now. It's in that other program. It's in that program, and then you got to paste it. I mean, before you do that, you got to
Right.
paste
All you
it.
have to do is just come here
Right.
and then just click on it, and it says copy to clipboard, and then you paste it to whatever tool
Oh, I see.
or Office program you want to use. So like if
What's
I-
view read support? Is that doing?
Oh, that's BAM file view again. When you load the BAM file, and you can see the reads aligning.
All right. I think. What's all that? Why is it red and green and blue up on top?
So this breaks it into your heavy V461. Here's your IGA.
Oh no, I mean above that.
Oh, this is just the color scale. So A's are a certain color, C's are a certain color.
Oh, that's ATCs.
So this is just their
Oh, I see.
ACTCs up there. Because if I zoom in by holding down my mouse button, I can zoom into the actual sequence and say, oh, here's a mutation, here's a mutation. So A's are green.
And you can slide that little slider along.
I can slide it along.
I was thinking slider up in front. Of course you can.
You can do that too. I bet you could do it. Nope. Oh yeah, you can. It just doesn't.
Okay.
So yeah.
All right. So, can you analyze the D region? I know this is like I didn't want to actually analyze the D region, but maybe it's useful. Is there a way to just sort of get the sequence and see how that compares to the database of that D? You know, there's a D there, and I don't even know what D it is, but
So.
what's an easy way to do that?
And it tells you what D gene it is.
Right.
Um,
So we want to say, okay, that's the D gene that it came from, but how many real mutations are there or whatever, and what this cell has?
in IGHD.
Yeah. No, no, the diversity region. I guess it's not the
That's not the CDR3, is that?
Yeah, CDR3, right?
Okay, so it's right here.
Okay.
And
Right.
so, okay, that's what I thought you were talking about.
Oh, okay,
My bad.
No, you're right.
So here's the CDR3 region for the heavy chain.
Right.
It gives the amino acid and it gives the nucleotide.
Oh, okay.
And
that's
supposedly
now, how
you
does
can
it?
click this link and say find like CDR3s, but I haven't had any success with that lately.
Now, can I ask you that CDR3 just came up right when I wanted it to? How did that come up? Did you move that slider?
No, I didn't, because it's always present at the bottom.
Oh.
This is always going to be at the bottom. Whatever barcode you search for, whatever thing you click on, you'll see it changes to whatever I'm looking at.
You mean
So-
everybody's instant CDR3?
Yeah, so this is the CDR3 for this particular heavy chain of that particular barcode.
Does it come up with a light chain? They have a CDR3 region too, right?
Yeah, I guess.
But anyway, okay, let's stick
The
with
only
that.
one that had a light chain was this barcode over here. It was this one.
Oh, I noticed that. I was gonna, but
It did not have a heavy or a kappa
Hmm.
were associated with it. There was just the light chain for that one.
that doesn't sound right, actually. Maybe maybe this all
Yeah.
just.
I don't know. I
Some-
mean, that's just, and maybe that wasn't sequenced deep enough, or it wasn't sequenced enough. I don't
Yeah.
know. It's possible.
That's interesting because we wouldn't pick that one. Of
Right.
course, but-
And some of them only had, I had both. This one only had the kappa
right.
and didn't have the heavy.
That's interesting.
And I still can't find this one in any of the data. So I
That-
don't know what that is.
Oh, you can't find it at all.
I can't find that barcode at all. Even in the rawest of the data, it's not there. So, I mean, and I double checked that I didn't type it in wrong because you had a picture image of all those,
Yeah.
and I just typed it out.
And
I
we
don't think
and we had a picture of the heavy and light chain too.
It'd be interesting to see
So
what it is.
I mean, it called it. Maybe I don't know. We have to go back
Yeah.
because that we got at a crude level. But what did you say about the CDR3? Can you sort of okay? So you got it there. I mean,
Well, it's even up here too. It's in the Excel file I sent you. Here's the amino acid sequence, and here's the nucleotide sequence of what that is right there.
now I'm saying okay,
Yeah.
that's CDR3, but you've done it before. What do you use to sort of? We've done it too. Put the CDR3, and you see the addition and the
Right.
hidden region and so forth, and you put it into a program, and then here's what it is, and here's the thing that changed, and that kind of thing.
Yeah, I don't know I know I've been intrigued with this button, Find Like CDR3.
Oh, there you go. Okay,
Um,
try that.
but it says filter the clonotype list to CDR3s with this chain's amino acid sequence. If I click on it, it now switches this over to CDR3 amino, and it takes that amino acid sequence and puts it here and searches the entire list of everything that has been sequenced in your samples, and it's still the same one sample.
Oh, I see. It's only going to work with that.
Right, it's
sample.
only going to look inside of the file, I guess, is how that works.
I mean, we used a data. We used a. And
but could you not take like the IGMT database or something and
Yeah.
put that in there, and
That's what I have done. I forgot it was like 10 years ago.
or IMGT, whatever the name of that database is for immunoglobulins?
Yeah, so, but I'm sure we can find it.
Yeah, I'm sure all you have to do then in that respect is just come down here and copy it, because it has the copy feature, or open up my Excel file and it's there too, and just copy it and then paste it into IMGT or IGMT, whatever the name of that.
now the
that
CDR,
program.
because what we found was also okay. So we'll work on the CDR3.
Mhm.
What we found in the BD2 was the IgG, the mutated ones, were mutated outside of the CDR3 region too.
Mhm.
Not as much as in the CDR3 region, but
Okay.
there was some, and I think I saw it. So how about that? I guess we know how to do that, right?
Yeah, it should be all there.
And we did it before, and and the good news is we don't have to do it for 2000. I mean, maybe we could do it for 2000 if it was automated, but we can do it for one and then get good at it.
Right.
And some people that's all they do, like Harry Schrader. That's it. His whole world is CDR3.
That's true.
But we don't, and so I talked to him actually 10 years ago, 11 years ago when we were trying to do it, and he said, and that was he was helpful, but somehow he didn't come to any conclusion. It was sort of amazing. He said, "But can't you just, like that's what I'm asking you. This
Right.
is all worked out, right? I mean,
Right. And so, I mean, this sequence down here shows this nucleotide sequence. So here's your CDR3 region. And so it starts with the TGT. You have this large deletion. Then you pick back up, and then you have a C that's supposed to be an A, another deletion. There's two G's here when they should be CT. So you have the sequence here all the way to the... How
IG.
far does it go? I can't remember how far it goes into this region.
So David, when they found it, since those are all those thousands a day, so the deletion we think that's real. It's not a sequencing.
Yeah, it's maybe not a sequencing error. Let's look at the read.
yeah, that's the
support.
thing. That especially that big deletion.
I go right here. So if I'm only interested in this sort of region, that's where the CDR3 well, no, that's not it, is it? Color coding is not. Well, I think that's it.
I think the green should be CDR3, right? That's the D. Well, the both.
I want to go
combined.
from the V461 to Yeah. Okay. So let's just see.
Maybe the J chain is missing or something.
Yeah, I want to go from here.
Or no,
to
D
there.
is next. And
Yeah,
then
this
J.
is where I want to be at.
Right.
That's where the CDR3 sits right here. Let's see here. I'm going to make this big.
Did it call a D region? Did it say it's D something or other?
Mm-hmm, it did. I'll show you where that's at.
I think this is the challenge for, this kind of sequence. Single this 10x. To me, it's a challenge because it's easy if it's not 10x, but other, because then you got to clone. But for 10x, you don't have a clone, and you just have these overlapping sequences. It's interesting. They sort of a lot of them start right here. The few that go through that region.
Yeah, and it might be a little weak. I need to pull that sequence and just have a little image of it so I can see it. Because I think it's this sequence here. It starts. Go back. Sorry.
Now, Greg next week should be helpful on this, right? Because he's Mr. Heavy and light chain.
So when you come over here and you hover over, you do see which gene they're being used, which V gene, which D gene, which J gene.
Right, so it thinks
so
it's
forth.
there.
So it's,
Where did
it's
you go?
clearly
How did you-
showing there's a large deletion of 18 bases here in that CDR3 region. Now I'm wondering if the reads, I need to look at it. I need to look to see how it's reporting that, because I did not see that deletion, but maybe it's using this as a reference now. I have to go look.
Can you, does it like that for, okay, so this is maybe, okay, we look at this and I'm not sure. So we might not clone this one, but we'll have maybe start with 20, 10 might drop out because they can't find the V or the something, and then blah, or light chain or whatever.
Right.
And then 5 more might drop out because
Right.
we're not sure. So that's what we'll do, and we'll get the ones. Now, so can you look at another heavy chain and see if it also looks like it might not be right? I mean, this is new territory,
Right.
I think, for
Well,
almost
let's go to
every.
the next barcode.
Right.
So the one that you had listed in number 2.
Because fortunately, we don't have a shortage of sequence cells, but
Search. Okay, so here's the heavy and kappa. For that, if I click on the heavy, again, you've got a large deletion of 29 bases there in that region. Here's the actual nucleotide and amino acid sequence of that. It's the only one out there of ones that have been sequenced in your 165 sample that has that amino acid sequence for the CDR3.
maybe we can look at. I mean, there is a way. I was going to ask this, but look at all of the heavy chains, and sort of get an overview. Because we just picked the ones we thought based on phenotype of the cell, but like they have this BAM file, right?
Mm-hmm.
So is there a way just to like let's suppose we were completely ignorant of everything, and we just want to look at all the immunoglobulin genes, especially these B cells, like that. Something we don't. I don't think that's it, but let's look at all the V regions, and we could do it like that and approach it the other way, and go in from, all right, let's look at arginine mutations or whatever, in the CDR3 region, and those might be ideal.
So this gives you, here's the CDR3 abundances for whatever, I'm right now picking IGH, you could do kappa or light, and so there's one that has 6, and it's got this particular frequency, and if I click on that, it leads me over here to this group.
Is that one of these 6, or is that everything?
No, so these all here were only represented once in the entire, had only one barcode associated with them. This one here, this particular CDR3 had 6 barcodes associated with it. But all of these up here that you had listed, or in that screen grab that you sent me, was only seen once.
Oh, okay. That's
So it's
so we
very
may
rare.
not want to get that one. I mean, if we had our druthers, we could pick one that has at least 2. Can you look at that one?
This
Ye-
6 one.
Yeah, the BAM file for that.
So here's the heavy chain, and so here's all 6 of those that we're seeing.
Oh, all 6. Okay.
Yeah.
And they're all the same.
Yeah, so they all look roughly the same.
Oh,
I mean,
sure.
there's a couple of mismatches here that aren't in the others. They all appear to have this same deletion region. Same thing here. If I were to, oops, wrong button.
Now the deletions in it looks like a different spot. If the yellows,
Yeah, the yellows are shifted over further. Or the purple's shifted over further to the end of the
Right,
CDR3
right.
region, where it starts from here and goes across.
Always a deletion though, with the P when it goes out. It's
Really?
when it's rearranging.
Is that right?
Yeah.
what was that?
Why during rearrangements?
Yeah.
The VCR, there's usually deletions in there.
That's why. It's
Well,
so
there
variable.
could be in region
Yeah.
addition too, though,
Well, that's
right?
true. You could have insertions. Yes.
You can have, so it could be. I mean, some people, Harry says, oh yeah, in region additions are critical for autoreactive ones, and the arginine mutation. So,
So yeah.
Yeah,
I mean, looking at all.
but I'm
6
looking. I'm
of-
thinking of deletion, but a short one. You this and that, but you don't delete like 10 or 20. I don't know. Maybe you do, but or maybe there's a problem with this.
Well, like in this instance, all 6 of these have this, C mutation. They have
yeah.
these 3 deletions, and then now there's an insertion here. Looks like of 1A across all of them.
And where is that? Where
So
is
it's
it?
this little blue line. You have to hover over it to see what the insertion is.
Oh, okay. I'm just wondering. I can't quite read it. Is that the D region?
Yeah, so this is CDR3. So CDR3 starts about here and rolls into that.
I see.
And
Oh.
So,
So that's but
and then you have this large deletion here. So, the CDR3 region's in this dark gray area.
so big deletion again.
Yeah,
Is
so
that
you have a
in the-
deletion at the beginning of 3 bases, brings you an insertion, and then a deletion.
Is that between the D and the J? Is that what it is?
Yes, that's correct. Yeah, so if I scroll back, this is the D33, and here's the J4.
I wonder what that means. I mean, it looks like there's always these big. Now they move around.
Right, they do. I mean, the other one we were just looking at that had 1, the deletions were all right here at the front of the D. So-
Like, is there some that don't have the deletion? Like they can't all be so mutated.
Mhm.
Is there a way to look at? I mean, I know there is. Like, what sequences do they? It seems like it likes to start. I mean, that one had the deletion. It was sort of like it's started. All the sequences came from the start position. That's sort of after the deletion or something like that. Like there's a problem with sequencing through that. And then, you know what I mean? And then, of course, it would say, okay, there's nothing back there, and the other said, well, nothing up here, and let's call it a deletion kind of thing.
Well, yeah, I mean, majority of the times, if it shows a deletion, it's like the read has these sequences, and then the read picks back up at this, and it doesn't have these bases inside of it. So there's an actual complete deletion.
And a
See if
read.
I
I
can-
mean, like you said, the ones at the beginning and end are not. But so is that in the middle of a lot of them, or?
See, the mutation was near the end. I don't think it's showing the mutation in this report. I don't know what that does. See, I need to look at how they've implemented IGV into their tool system, because I have a feeling that what they're showing here is not the reference, but it's the actual sequence of the thing, not of the reference. And I'd like to have, and when I open up IGV, I have IGV installed locally on my machine. You can just put a BAM file directly into it. It will have a reference, and then it will have your reads and all that aligning to it, and then you'll see that deletion. You'll see that the read, I wish there was a good one in here, I just don't see one.
What are you looking for?
I'm looking for a deletion, and I don't see it in this data, which tells me that the way they have their IGV set up inside their package is that it's not going to show you, this is not the reference.
What do you mean it's not? It's not.
I'm guessing that's not the actual reference in the human genome.
Oh, okay.
And
That's
what it
actually a
is,
sequence.
Is the actual sequence of, come on, it's that sequence.
Oh, okay.
And I need to sit down and I
in
need
that
to copy
sequence-
that and then look at it in their IGV to see that it's.
So you're saying that sequence doesn't appear to have that deletion?
Well, it shows it here.
Yeah.
But when I then want to go and see the reads that support this,
Right.
I don't see that deletion, and that deletion should be right here.
How would the
And-
deletion show
Well,
up?
the deletion will show you. It would be like you have this red rectangle here,
Yeah.
and let's say the deletion was right here. That red rectangle would be split,
Oh,
and there would
okay.
be a line like that in between.
So do you see one that goes through that?
That's I haven't seen one at all
Down
in the
there.
status. That's what tells me that I think this is not reference to the human. It's the actual sequence
Right.
of this particular barcode.
But somebody down there should go through that area, right?
Something should. So like that one goes all the way through, which tells me that I should see a deletion mark here, and it's not.
Mm.
So again, I still don't know what the colors of these different ones are.
That's interesting because now I can start to see. Oh, what's that? What's that gray graph above? Is that the number of reads?
That's the depth. So
Yeah.
you see depth of coverage is really high here at the 5' end, and it starts to tail off here. And you don't have very much coverage out here in the J and the M, the heavy M.
Would it help to sequence deeper? You know, because I can see the problem now. But now it's obvious that this approach is going to bias reads, of course, towards the 5' end, which is not exactly what we're interested in, and then we're going to piece it together, like the guy explained a long time ago.
Right.
But it's not going to have many representatives from the VDJ or the CDR3 region because it's too far down.
But shouldn't it? Because that's the way they design their or they design their
Well, they
their
all.
kit is that you can see the CDR3 region. The gene expression is 5', but I didn't think that the B cell receptor VDJ stuff is a 5' type assay.
I have to
But it
think
clearly
about it.
looks like
Yeah, it looks like
because
it.
the way of all the reads, but it should be a full coverage across every VDJ.
Yeah, but you can see the read.
And maybe something was not done right in the kit. I don't know. I mean, that's something to look back at it. The thing. And
I mean, the
do-
way the guy explained it to me, it's the way the guy explained it to me is it's going to start at the 5', but it'll find some other ones that are. I forget whether it's chopped up or something. I forget how it found things additional, and then it was going
Right.
to splice them all together.
And so,
But
we did paired in sequencing. We did paired in 150, and that's what they recommend. And so, an example to see what a paired in looks like. So this is read 1 here, and then you have a line that separates to read 2. And so these two were sequenced like this, and you had a small insert fragment.
And the other one below it sort of told you.
Right, and the other one here looks like it was just all one. Here you have another one that is paired in for sure. You can see the nice one. Others you don't see any sort of like this one's a lone mate. Its read
Right.
2 was who knows where, and that would have to be it.
Now, is that like 100? Is that I can't read the scale. 50, 100? Is the length of the red one sort of the common length? Is that like
Yeah, that
100?
should be 150 bases.
Okay, it does 150
be
bases at a
150,
time
yeah.
or so.
Right. Well, that's all we sequenced was 150 bases in the forward read and 150 in the reverse. That's those FASTQ files you got. They're 150 bases in length.
Can you do
That's-
more? Can you do it longer?
Yes, you can. But I think we were following what 10X suggested. They said to do paired in 150. And that's maybe we need to go back to their protocol and their kit and see. Well, is it a 5' bias? It looks like it is. But
Yeah.
To my knowledge, I didn't think it was supposed to be. I thought it was supposed to sequence the entire VDJ, and I would expect to see a nice coverage histogram here across all of
Right,
it.
but you can see it
But I
tails
don't.
off. In fact, I don't see any gray going
Yeah.
to the
Right.
CDR3 region.
Right, and as you scroll down, you see that most of the reads are over here on the 5'
Right.
end, and that you don't see very many at all over there.
I mean, something to, because nobody's done this,
Right.
I'm pretty sure.
Yeah.
And now we get the bad news that it's really 5' bias.
So it says there's 199 reads, so 200 reads depth coverage at least on the 5' end.
Yeah. Well, we'll
So,
ask Fred.
Yeah, you need to ask the 10X folks. I'll go back and look at the protocol and what they say.
We can do this, but can you send us a snapshot of that just in case we never get around to finding it?
Yeah, yeah.
Because I was asked Fred, look, it's 5' bias
Right.
here, obviously in the CDR3 region.
Well,
is-
it's clearly 5' bias, and this is the one with the most barcodes
Right,
in
so
your whole sample. So
Can you
yeah.
you send that snapshot just in case? This is an easy question, but it looks like they say, "Oh, I didn't know that" kind of thing, or did you read more or whatever? I don't know what the thing is, but we.